Good afternoon, dear students. Today we are going to study a new topic, that is scale up of fermentation process. Last time we discussed about inoculum buildup. Okay, scale up of fermentation process. Why scale up is essential? See, one of the most important and complicated aspect of industrial microbiology is transferring a fermentation process from small scale laboratory equipment to large scale commercial equipment, which is called as, the process is called as scale up. That means transfer of a fermentation process from laboratory scale to commercial production scale is called as scale up, right? It is also extremely important to understand the problems that we face during scale up, because microbial processes rarely behave in the same way in a large scale fermenter as in a small scale laboratory equipment. One thing we need to understand is controlling a microbial fermentation or a microbial process in a small scale lab fermenter, in a laboratory scale fermenter, or in a flask is very easy. But controlling the same fermentation on a large scale in a commercial pilot plant and or pilot plant fermenter is difficult, and hence we need to understand the scale up studies. Such problems arise because mixing and aeration can be easily accomplished, or it is very easy to carry out mixing and aeration in small laboratory flask than in a large industrial fermentation. It's easy. Aeration, mixing, everything can be easily controlled in small lab flask than in a large industrial fermenter. Further, the surface to volume ratio changes with the increase in size of the fermenter. In the large fermentation tank, volume of the liquid medium is greater for a given surface area. In a small tank, surface area is more, volume is less. But in a large tank, volume is very much greater as compared to the surface area. And gas transfer and mixing depends more on surface exposed than on fermenter volume, and hence it is difficult to mix the contents of a big tank in comparison to a small flask. Okay, what I said, gas transfer and mixing depends more on exposed surface or surface area than the fermenter volume. In a small flask, the fermenter volume is less, surface area exposed is more. So mixing, gas transfer, oxygen transfer, everything is very easy. But if you look at a large fermenter, the volume is more, the surface area exposed is less, and hence gas transfer and mixing is difficult in a large tank. Further, oxygen transfer is much difficult to obtain in a large fermenter, again due to surface to volume ratio. But oxygen transfer is very essential since most of the commercial fermentations are aerobic, and because the process is aerobic and aeration is reduced even for a short period of time, the culture may experience partial anaerobic condition, which ultimately affects the final yield of the product, right? So oxygen transfer is very essential because most of the commercial fermentations are aerobic, and if aeration is reduced or obstructed even for few seconds, few minutes, then it may hamper the product yield. If you look at a large fermentation vessel, there are packets. There are areas in a large fermentation vessel where mixing is less efficient. And if microorganisms are present in such areas, they experience different environmental condition than they would experience in the main bulk of the tank. Such packets do not exist in small lab flask. Okay. So, there are chances that microorganisms in a large tank may come across anaerobic conditions. Thus, the determination of proper incubation conditions to be employed with large scale production tank as based on the information obtained from various small size tank is known as scale up. So what is scale up? Understanding, determining the proper incubation condition, the media design, the pH, the temperature, everything which is to be employed in large scale as based on the studies or information obtained from small scale tank is known as scale up. So it is the task of the engineer to carefully handle the scale up of industrial process. Since the engineer is familiar with gas transfer, fluid dynamics, mixing, thermodynamics, etc. In case of scale up process, the industrial microbiologist works closely with a biochemical engineer to ensure that all the parameters that are needed for a successful fermentation are understood and all parameters are made available. Okay, and also the microbial strains required for large scale productions are also made available. So all these things need to be taken care of. The instrument, the fluid dynamics, gas transfer, mixing, etc. is very important. Very often it is found that the microbial culture medium which works very well in a laboratory flask does not work well in a big fermenter. Okay, the same culture medium, if you use it on a small scale in laboratory, it will give you good results, good product yield. But if you use the same culture medium on a large scale, you won't get a expected yield. That means very often the fermentations behave in a different manner on a small scale and on a large scale. Hence, a fermentation process, a production cannot be directly transferred from small laboratory flask to a large scale commercial fermenter. Okay, because there is a vast difference in the volume, the requirements, the behavior of microorganism, etc. So, we cannot transfer a process from small laboratory scale to large commercial scale production. So, what we have to do then? Slowly go on increasing the volume of the fermentation medium. Slowly go on increasing the volume. Also study various parameters and optimize the conditions that are required in a large scale commercial fermenter. So when you are transferring an industrial process from laboratory to commercial fermenter, several stages can be visualized. You cannot directly transfer a process from laboratory scale to large commercial scale. In between there are various stages. What are those? Number 1, you have to perform experiments in laboratory flask, which is generally the first indication that the process of commercial interest is possible. Okay, so the first thing you do is study whether a desired product can be obtained or not, and that can be done on a laboratory scale. And if you get good results in laboratory scale, it is the first indication that the process of commercial importance is possible. Right. Now from lab flask you go to a small scale fermenter. The laboratory fermenter, a small scale fermenter generally made up of glass having a capacity of say 5 to 10 liters, in which the first efforts of scale up are made. See, actually scaling up means what? Going on increasing the volume of the medium and hence going on increasing the product yield. Okay, but not an abrupt increase, a slow and gradual increase in the volume of the medium. So if you have an early May flask of say 500 ml, 250 to 500 ml, where you have studied whether the product formation is possible or not, and if you get good result, you transfer it to a 5 to 10 liter tank. Okay, that means you gradually go on increasing the process or volume of the medium. Okay, it is possible to test variations in the medium, temperature, pH, etc. inexpensively without too much of cost in a laboratory fermenter, since little cost is involved in the equipment or culture medium. Okay, so from laboratory flask you go to small scale laboratory fermenter having a capacity of 5 to 10 liter, where you can try all variations, go on changing the media components. You can bring about change in temperature, pH, and carry out studies or optimize the process. Optimize the temperature, optimize the pH, optimize the media components, and this all you can do in a lab fermenter inexpensively without any too much of expenditure. Right. After laboratory fermenter, we go to a pilot plant stage. Then comes the pilot plant stage, which is usually carried out in an equipment having a volume of 300 to 3000 liters. The conditions in this pilot plant vessel closely resemble or closely approach the conditions that exist in a large scale commercial fermenter. However, cost is again not a major problem in pilot plant stage. Okay, in pilot plant stage, cost is not a problem. So, but the conditions that exist in a pilot plant stage fermenter are almost similar to that which exist in a large scale commercial fermenter. Okay, so pilot plant stage is important. Careful instrument selection, computer control is desirable in the pilot plant fermenter so that conditions most similar to those in laboratory fermenter can be obtained. So after lab fermenter, you transfer the process to a pilot plant stage having a capacity of 300 to 3000 liters and study the process in detail. And then you transfer the process to a commercial large scale fermenter having a capacity of 5 to 10,000 gallons or even much more capacity. It is generally found that in scale up studies on aerobic fermentations, stirring, that is mixing, and aeration rates are kept constant as the size of the fermenter is increased. That means mixing, the rate at which mixing is done, the rate at which air is pushed in or air is pumped inside, that means aeration and agitation are kept constant as the size of the fermenters go on increasing. Right, so in scale up studies, mixing, aeration, agitation is all need to be taken care of. Thus, scale up of fermentations play an important role as far as the success of a fermentation process is concerned. It allows you to study various parameters that are required for optimum and effective growth of microorganism and production of a commercially valuable product. Right? So scale up studies are important. We slowly go on increasing the volume of the fermentation medium and hence the volume of the media that you use, and you go on increasing the production. Okay, and when you go on stepwise increasing the volume of the medium, you actually are transferring the process from a small lab flask to a laboratory fermenter, then to a pilot plant scale, and make sure that all parameters can be controlled. The product can be successfully obtained from the substrate, and once you are sure that this process is possible on a large scale, then only proceed for large scale commercial production of the product. Otherwise, it will cause a huge economic loss. So to avoid time loss, economic loss, we have to study scale up process. Okay, so if you look at the diagram, you can see a small laboratory scale fermenter, then a pilot plant fermenter, and then a large scale commercial fermenter. This is how the process is transferred from small vessel to a large vessel. Like lab scale 5 to 10 liter flask, then 300 to 3000 liter pilot plant stage where all studies are done, and then finally transferring the process to a commercial fermenter which may be having a capacity of 10,000 or more gallons. Right, that is all about scale up of fermentation process.